started. This is Behind the Scenes of Science. My name is Caitlin. The goal of this webinar series is to give you a sense of some of the careers in science, different pathways. How do you go from being a high school student to being a person who works in the field of science? And what does a day in the life look like if you work in the world of science? So today we are talking to Greg, who works in the immunology division at the Allen Institute. Again, I'm going to start with some questions and then open it to questions from you. So Greg, if you want to start by introducing yourself.
Sure. Hi, everyone. My name is Greg Szeto. It's my pleasure to be here and chat with you all today. I guess I can start with my background. So I grew up in Waldorf, Maryland, a pretty rural suburb of Washington, D.C., about 30 minutes outside. And I guess my journey with science and my career kind of started pretty early. I was super interested always as a kid in technology, new gadgets, Legos, and Transformers. I would say the rest of my career looks linear on paper, but it was actually highly nonlinear in terms of I had a lot of detours and challenges along the way that I had to kind of grapple with. I'll tell you a little bit about some of them. Happy to answer more questions about any specific points. And so I guess my professional science journey actually started in high school. So around my sophomore year, my counselor actually suggested that I should apply for this internship, summer internship. And she said, what are you interested in? And, at the time, and still, I'm super interested in computer games. And so I said, something to do with programming. And so I got dropped into an Army lab that was doing programming, looking at processing of sound. So taking the recordings from like drones and figuring out like where troops were moving in different places using computer programming. That was super interesting. Didn't really totally mesh with my kind of in-school interests, which were mostly chemistry. I actually really hated biology in high school. That's a long story actually. But then when I got to college and where I went to college in Carnegie Mellon in Pittsburgh, I kind of went there for two different reasons. One is I had a strong passion and have a strong passion for music and sound, as I indicated for my summer internship. And so I wanted to go to a place that had as good science and engineering as it did fine arts in case I wanted to drop out of engineering or science and go be a tubist. So I actually did both for 2 of my 4 years there. I was a triple major tuba, biomedical engineering, and chemical engineering. Biomedical and chemical came a little bit later in my career. My initial degrees were actually computer science and chemical engineering because my thinking at the time was, I love computer games, I like programming. How do I merge that? Well, maybe I can make faster, better processors so that my games would run faster. We could make more realistic games. Somewhere along the way, I got distracted by medicine. I remember one talk specifically about gene therapy and a clinical trial and death due to response to the virus. I got super interested in biology in general, not kind of the biology that I learned in high school, but the biology that was more translatable and molecular and specific to humans. And so I started going down that path by actually switching from computer programming to biomedical engineering. And I did an internship in early bioinformatics, figuring out gene sequencing, how to connect gene sequences to different parts of the genomes of flies actually at the NIH. That led to a biomedical research position at University of Pittsburgh Hospital, where I worked on gene therapies for muscular dystrophy. And that was a really cool experience, and that kind of cemented where I wanted to go for grad school. I was about 50-50 of going to grad school or going to industry for chemical engineering because you make a lot of money coming out of a bachelor's with chemical engineering. But I decided to go to grad school because I really wanted to become more of an expert in immunology, and specifically the immunology against viruses. And so I went to Johns Hopkins School of Medicine and got my PhD in HIV immunology and how HIV interacts with the immune system. After that, that was a super deep biology experience, and I was missing my engineering side. And so I actually did what's called a postdoc, or so my after PhD training period up at MIT, where I actually combined my interests with immunology with my formal training in engineering in what's called an immune engineering lab. And so there I worked on nanoparticles, I worked on mathematical models, I did a whole bunch of stuff in cancer, HIV, vaccines, tech development, software development. And then I actually was, around 2015, I joined the University of Maryland and was an assistant professor in chemical engineering there. So I did that for up until March of this year when I actually joined the Allen Institute for Immunology. So here I'm an assistant investigator.
That is a great overview, and I think that's going to spur some questions from our audience about the different stages of your career. But now that you're at the Allen Institute, what does an average day look like for you? And normally I would ask, what does it look like pre-pandemic? But you joined us right at the start of the shutdown. So what does it look like, an average day look like for you now? And what did an average day look like for you back at Maryland?
Sure. So let's start with the past, because happier to think about in some ways. Back at University of Maryland as an assistant professor, I was doing the things that a professor does, teaching classes, helping run the department and running my own research lab. Mentoring graduate students, teaching undergrads how to do research for the first time, writing papers about the science that we did, writing grants to try to get funding. Now my existence is obviously very different. I joined the Allen Institute actually right as lockdowns were getting started. And so I've been pretty much work from home. As you can see, this is my daily experience. For the most part, I still get into the lab occasionally. That was part of the bargain of joining the Allen Institute, was that I would get a little bit of time to go back into the lab and do some developmental work. And so now my days actually look like, so there are a ton of online meetings, webinars like this, because now I don't spend so much time in the lab anymore. I'm more kind of thinking about the science that we do, strategically designing what science we go after, figuring out what are the interesting questions that about the human immune system and human disease that we can try to address with our resources and our research.
And what is one of those current research questions that you're working on? Something that
so the primary one I'm working on right now is actually COVID-19, SARS-CoV-2, and specifically with Fred Hutch and Dr. Julie McElrath's lab. Our institute is in a collaboration to figure out what the path is for people who are asymptomatic or mild in terms of their symptoms from infection to immunity, and what that immunity looks like and how it performs over the course of 2 years. And so that's called what's called a longitudinal study, where you take the same person pretty soon after they were infected, grab their blood a whole bunch of different times and test them with a whole bunch of really cool assays, tests in the lab. And so another part of my day is actually trying to figure out how to deal with what's called big data. So like terabytes and terabytes of data about biological systems. And, I have a lot of time on the computer now programming in, for example, R or Python, doing statistical analyses and computational modeling of our data.
Yeah. Those of you in the audience, if you have questions about this research, in addition to what it's like to be working on it, please go ahead and put those in the Q&A. We'll come back to those in a minute, but before we talk about that, what is your favorite thing about your job?
Ooh, that's hard. There's a lot of things that I like about my job and also the Allen Institute in general. I would say the top thing that I like about a job and career in science in general is that I'm never bored. I get really distracted really easily. And so, for example, I'll give you an example. When I was running my research lab at University of Maryland, we focused on topics spanning from traumatic brain injury, cancer immunotherapy, lupus, and cellular metabolism, for example. All as it has to do with immunology. As long as there's some component of the immune system, I'm usually pretty engaged. So it's hard for me to get bored with immunology, which is pretty awesome to be in the Institute for Immunology, because that's all we do is immunology. But I like to bring in lots of approaches and tools from other fields like engineering. So thinking about, creating 3D mimics of organs in a cell culture dish in the lab, or new algorithms for data processing, that sort of stuff. I would say kind of a corollary or related to why I don't get bored is because I'm always getting to think about new exciting things. And so I think a cool part about a career in research in science and engineering is you're often asking questions that nobody has asked before or that nobody has answered before. And so that thrill of like finding the answer to something unknown or unknowable, previously thought to be unknowable, through technology, through your own ingenuity, through the team's ingenuity, that's really, really enthralling, I would say.
Yeah, there's really nothing like looking at a finding and realizing you're the first person to know this.
Definitely.
Yeah. Well, we have a couple questions from the audience. Let's go ahead and tackle those. So we have a question from David about the COVID research. Are you looking at changes in protein shapes post-virus? Or I guess broadly, what are you looking at in the immune response of the people who are participating in this study?
Yeah, so we're taking what's called, sometimes termed a systems immunology view, which is we try to take a holistic view of the system of your immune system, which is, all the cells, white blood cells that run through your blood and, crawl all over your organs and help protect you day to day, right? And so how we dissect that is we think all the key biological molecules that, you all should have learned about in basic biology: DNA, RNA, protein, right? So DNA is your genetic code. There's a sequence to it. The first thing that we look at is that. The second thing we look at is actually there are chemical modifications to DNA called epigenetic modifications. And those can actually directly change how DNA functions and translates into, transcribes into RNA. So DNA to RNA is changed by chemical modifications on the DNA, and we look at those chemical modifications and where they are in the DNA of people. We then look at RNA, and then we look at proteins. So we don't look at the protein shapes per se. So shape is another word for what's called, more technically in the field, conformation, I would say. And so we look more at abundance and activity. And so abundance is how much of the protein there is. Activity is what is it doing? Is it active? Is it inactive? So abundance would be like, how much antibody do I have against the virus? Function would be like, how good is this antibody per molecule at, say, binding and preventing a SARS-CoV-2 virus from infecting a cell? So that's one of the tests that we use is called a neutralization assay. So you take antibodies from the blood of somebody that's been infected or who has gotten a vaccine, and then you incubate it in the lab, mix it together with some virus, and then you look at how much virus is able to infect after that incubation.
And that'll be happening for 2 years post-infection, is that right?
Yeah, up to 2 years.
The study, that is.
Yeah, we're working on our first paper of our findings right now, where we're describing kind of the first few months of infection to the first signs of an immune response. But, we're hoping to follow out to 2 years, both natural immunity as well as vaccinations. We would expect that most people that have gotten infected would get a vaccine.
Yeah. Well, going back to an earlier stage of research, we have another audience question. Oh, we have a follow-up on the COVID study. What is the reason for a longitudinal study?
Yeah, so that's one of the underlying hypotheses or ideas, that concepts that kind of fuels our institute, is that each person is an individual. And so in a lot of studies in science, you'll often take a bunch of stuff that is actually, when you look at it under a microscope or through a more detailed lens, there's actually a lot of different things in that, in a sample. For example, in your blood, there's, millions of white blood cells. And then in those millions, there are, dozens and dozens, if not hundreds, of subsets of cells that each function differently. So one of the ways that we pursue research that's relatively unique is what's called single cell research. So we take each individual cell and we interrogate its, chemical modifications to DNA, its levels of RNA, its levels and functions of proteins. And so by having this more detailed view, it's kind of like taking each of the Skittles from a bag and like dissecting it rather than just looking at a bag of Skittles without opening it, is maybe one analogy. And so to take that further though, a longitudinal study would say, okay, this bag of Skittles, as I'm eating it and then following everything as it's getting digested, to take it to its extreme, right? And so each individual person will eat the Skittles at a different rate. They'll respond to the Skittles differently. They might digest them differently, relatively speaking. And in the same way, we hypothesize that each individual person's immune system is unique to them, and how they respond to diseases, whether it's SARS-CoV-2 infection and COVID-19, or cancer, or autoimmune diseases, or even vaccines, or even just maintaining healthy, being normal, right? How do you define normal? I think that's a very individual answer. And so by taking a longitudinal analysis, what we do is we look at the same person before and after, or during and after a disease. And so we get a sense for how each person responds differently.
Yeah, there's longitudinal studies take a lot of time and effort, but there are things that you can get from them you just can't get any other way.
Yeah, I think particularly for humans, that it's a critical thing. And also, it's really dependent on people being generous in general in terms of recognizing the importance of scientific research, participating in trials and studies. I've participated in any number of them myself. So I think it's a really important thing to acknowledge also that it's not possible without willing patients, right? And especially when you think about some of the conditions we work on, like multiple myeloma, these are very severe cancers, and you're asking a lot of somebody with more than likely a terminal illness, right? And so it's really only because of people that we can do the work that we do.
Another question from the audience about some of your past research, which also is some disease research. At the University of Pittsburgh, you worked on muscular dystrophy. Can you talk a little bit about what you did there and what was that experience like?
Yeah, so we were working on Duchenne muscular dystrophy, and there's a gene that's specifically mutated in that muscular dystrophy called dystrophin. And what we were working on was trying to introduce that gene, engineer it into a virus that then you could give to a patient and inject in their muscles, and it would lead to regeneration of muscular tone and function. And so we did a lot of that in human cells in a petri dish, or using mouse models of muscular dystrophy, because that's one of the only ways that you can get to the function of the muscle is to examine it in a model organism.
Yeah, so audience members, we do have time for more questions from you. Please go ahead and put those into the Zoom Q&A or into YouTube chat. We are watching that side of questions as well. What do you wish you had known when you were in high school about a career in or around science, or about immunology specifically?
I wish that I knew that my failures to memorize things in high school biology would not translate to any sort of prediction of my success or failure later in life. I wish I hadn't written off biology as early as I had in some ways. Yeah, I would say there's a lot of excitement in biology once you get past the basics, memorization of things, which is, it's kind of necessary because you kind of have to know the general rules before you know all of the exceptions. And the exceptions are where it's interesting, and there are more exceptions than there are stable rules, I would say, but you just have to get there. But if that is interesting to you, push through the memorization slog for sure.
Yeah, taking a class in biology or whatever field of science is very different from working in research in that field of science.
Yeah, and that was something that was illuminated for me early on because I had a summer internship. So I would say, if you have an opportunity, even if you don't think you're competitive for something, definitely apply for it because you never know, what could happen. And so if you're interested in a career, potentially in something, my motto has always been to try it. So I can give another example, not related to science, but when I was a grad student, I actually ran a music blog in Baltimore at the time. And I actually almost quit grad school because I wanted to run my music site and do music journalism and photography. And so I think it's really important. I guess that would be my second thing I wish I told myself more, is that it's okay to have, lots of interests. And that you can still accomplish things and be successful and have a personality, diverse interests, hobbies. It's important to keep those up.
Yeah, so in addition to tuba and music in general, what do you do outside of work?
Let's see. I love spending time with my dog and my family. I am, I guess I would say, pro amateur photographer. So used to be concert photography exclusively. More recently, like past 5 years, I've expanded to landscape photography and kind of urban, like situational photography. Photography of, everyday life events and trying to capture moments like that. So I've been really into photography, still into music a lot. So.
Do you still play the tuba?
Yeah, I am teaching my 3-year-old daughter to play the tuba. It's pretty funny because she just kind of yells into the mouthpiece and into the tuba because, the mouthpiece is like as big as her, half of her face. So.
We have time for a few more questions from the audience. If you want to go ahead and put those in the Q&A. While you think of your questions, what has been your best science failure? Whatever that means to you. Best learning experience, funniest thing you've ever seen, most surprising.
So there's, and let's see, my biggest science fail was probably, I was working really late in grad school. And this was actually between Christmas and New Year's, one of my first years in graduate school. And I was working with HIV patient samples. And I was trying to figure out how much virus was in these samples. And so I had to do all of these different, like big stacks of plates and plates, like hundreds and hundreds of different samples. And I was like almost done. And then I took them out to the machine to read them, but then I was getting kind of tired, so I went and got some coffee. And then when I got some coffee and I brought it back, I grabbed the plates, and then my hand shook a little bit after, kind of guzzling a bunch of coffee, and all the samples went like everywhere. So like 12 hours of time and like probably 5 to 10 different patient samples were just like all over the floor. And that was a pretty big fail.
Oh, that's rough. We got another question from the audience. This is a great question. Is there anything unique that the Allen Institute offers in our approach to science versus other places you've been? And I think the COVID study is a good example as part of that.
Yeah, there's a lot of different things that are unique. I think one big thing is obviously teamwork. I wouldn't say it's unique to the Allen Institute, but relatively speaking, in science and engineering, teamwork is becoming more and more critical to do bigger science. And so the classical concept of like scientists kind of hunkered over a microscope trying to discover something on their own in their attic or something, that's extremely rare now, I would argue. And I mean, for example, just the COVID project, we need clinicians and nurses to go to the patients, to consent them for the study, to get their blood samples. We need people at the labs at Fred Hutch to process those samples. We need, couriers to deliver those samples over to us. And then we need our in-lab staff to do all of the complicated single cell analyses. And then we need our computational staff and our software staff for doing, taking those data and turning them into real scientific insights and knowledge. And so it's a huge team effort, for just one project, right? And so in our institute, we're doing, in the Institute for Immunology, we're doing the COVID project, we're doing a myeloma project, we're doing a melanoma project, we're doing arthritis, we're doing inflammatory bowel disease, and young and old healthy people. So that's 7 different projects. So imagine the teams needed for each of those projects, right? And so team science is huge and transformative. And so I think another aspect that's really important to note is, philanthropic funding, kind of the ability to do research and ask scientific questions without as many logistical and financial barriers is really important. So being able to formulate the best questions and pursue the best, most rigorous answers without funding restrictions is really, really amazing. Because typically in an academic lab, or even other nonprofits, you would often need to write grants and get the NIH to fund you. And so here we can also do that, but it's not necessary to keep the lights on and to keep research going.
And that in addition to working on these large teams that can tackle complex projects, and also being able to use this flexible pipeline of data analysis in the Institute, that you were able to turn that eye onto COVID very quickly. David asks, is this multidisciplinary approach of these teams working together becoming more common in science? And I would say yes. And
definitely, I think, yeah, I mean, I think my entire career has been multidisciplinary, interdisciplinary, or multi. Chemical engineering, biomedical engineering, biology, immunology, virology, single cell, computer science, data analysis, right? Those are all separate fields that people often will spend their entire careers in. And so I find it most fun to kind of be at the intersection. And so some of my mentors have called this type of research integrative research or convergent research, because it means you're pulling from multiple areas to create a unique solution or a unique approach by combining the strengths of all of the areas. And I think that this is exemplified by, so the Moderna coronavirus vaccine is actually a triumph of, I would say, bioengineering, as well as biomedical science and technology and basic biology as well. The mechanisms of how to make mRNA, right? How to package that into what's a lipid nanoparticle. And so lipids are the things that make up the membrane around your cells. So in the drug delivery field in chemical and biomedical engineering, these have been used for a long time to actually stabilize drugs and what's called encapsulate, or grab them into little spheres and nanoparticles for more efficient delivery and protection from being degraded or eaten up by enzymes outside of cells.
Well, we are just about out of time. Any last thought for our audience?
No, just thank you for the questions, and thanks for listening to my story.
Thank you everyone for being, thank you everyone, and happy end of 2020 and Happy New Year.
Thank you. Stay safe.
Thank you.